first session of the afternoon, which is molecular testing for biliary tract cancer in Canada. And we'll have 3 speakers, which I'll introduce, and then we'll have a panel discussion at the end as well. So it's my pleasure to introduce the first speaker, Dr. Harriet Feilotter. She's the division head of UHN Genome Diagnostic Program. And she leads as I mentioned, the Genome Diagnostic Program and Applied Molecular Diagnostics Lab at UHN. And she's the clinical lead for genetic testing at Ontario Health. She's an adjunct professor at Queen's University and co-leads implementation science at OICR. And through these roles, she advances education and research to integrate genomic biomarkers into clinical practice. And as anyone at C3 in Ontario knows, the UHN lab is supporting our C3 molecular testing program for patients in Ontario. So thank you very much, Harriet.
Okay, thank you very much, Rebecca, and thank you, C3 and everybody, for inviting us to be here. So in this session today, we're actually going to talk a little bit differently than we did this morning. So this morning you heard a little bit about molecular biomarkers and you heard about research in that area. What we're going to talk about now is a little more closer to what is standard of care or what's actually being offered to patients in across Canada. And in order to start all of this, I'm actually going to talk a little bit about the molecular biomarkers in biliary tract cancer. And then I want to talk about how we actually measure them. And I know that that might sound really kind of boring, but it's important to have at least a sense of how we measure these biomarkers, because quite often what we find is that the lab portion of what happens to patients is very much a black box, right? So from the clinical and from the patient point of view, often it's, well, and then we'll get some molecular testing done. Well, I'm in that black box. That's where I live. I'm actually a molecular geneticist. And so the black box is really important to me. So I want to talk about how we measure those markers and why that matters to you and to your clinician. And then I'll go very briefly through the Ontario experience. And then I'll hand it off to my colleagues to talk about Quebec and then the rest of Canada. Now I'm going backwards. I have no disclosures that are relevant to this talk. So you've already seen this slide before a couple of times today. So this is again just reminding you about some of the biomarkers that we care about because there are targeted therapies that are associated with finding mutations in some of these genes. So when you or your clinician maybe looks at this, you probably look at the far right column, which is the molecular therapy that is associated with having one of these mutations. When I look at it, I'm actually looking at the first column because I'm trying to figure out what is it that I need to be able to see with the assays that I'm using in my lab. And so I divided these into sort of the different types of mutations. And Dr. Grant this morning, when he spoke, he told you the DNA to RNA to protein paradigm. And we think about that all the time. And so I divide the mutations into the kinds of mutations that we care about. The first one's at the top, single nucleotide changes. So these are just DNA changes like misspellings in the protein coding genes. Those are very easy for us to handle. We usually use next generation sequencing or big parallel approaches because we can look at lots of these at the same time in a very small amount of tumor. And that's pretty easy. We don't really have to talk about that. The copy number is a little different. That's where you're talking about there's maybe more than one copy. Maybe there's multiple copies of a gene or a gene is completely deleted or a big piece of it. But for the most part, we can also see those using NGS approaches, so we don't really want to talk about that. What I do want to talk about is the fusions. So the fusions are kind of an important story, and they're kind of tricky to look at, and they're important to understand in cholangiocarcinoma in particular. So what happens in a fusion, and I'm sorry I didn't put a slide in to show you this because I didn't realize that there was going to be a lot of patients here and their families, so I should have done this, but I'm going to do it with my hands instead. So what happens when you get a fusion is you basically have a gene that is doing something. It's got a business that it's doing. And there's a break in the chromosome right in the middle of that gene. So it splits the gene in two. Now, this happens all the time in chromosomes. They normally get repaired again. But let's say it happens in the middle of an important gene whose job is normally to make a protein that controls cell division. Okay, so now you've got a break. And let's say there's a break somewhere else. And it's in the middle of another gene. And the other gene's job might be something completely different, but it's busy doing its job. And then when they come back, they join together by mistake. So now you've got 2 pieces of chromosome, each of them ending in half of a gene, and they've come together. So what happens in the case of cholangiocarcinoma is this gene FGFR2, you can see it the 3rd line down, that gene is a tyrosine kinase. So it normally has an activity to regulate cell division. And in a gene like that, you want to turn that gene off. You want it to stop making RNA and stop making protein at the right time because you don't always want to be signaling that the cell should be dividing. So FGFR2 is very tightly regulated, so it won't make RNA and it won't make protein at the wrong time. So what happens to it is it'll get broken. So now you've got this gene and it'll link to another gene that is busy making RNA and making protein for itself because it's supposed to. And what happens is it will now drive the business end of FGFR2 to make its protein as well. And now you've got inappropriate signaling from that gene, which is turning on cell division. And so that's one of the events that happens in a disease like cholangiocarcinoma. And it's one of the events that we want to be able to see. So I hope that made sense. So here's a schematic of the FGFR2 gene. You really don't need to see much of this. There's a few things I want you to pay attention to on this. This is a DNA level schematic. So what you're seeing is the little blue boxes are what we call exons. Those are the coding regions of the gene. And the thin lines in between are the introns. Those are kind of junky DNA that's between. And what happens at the DNA level is the gene looks sort of like that. And then all those blue boxes get brought together in the RNA. You lose all the stuff in between, and then the RNA makes the protein. And the thing I want you to see in this slide is that the little yellow bit at the very end on the far left, this side. That'd be the right? No, it's not. Okay, so I'm terrible that way. Anyway, is the tyrosine kinase domain. So that is the business end of this molecule. So you can see that the exons that are labeled 10 through 17, those are the things that make the tyrosine kinase part of the protein. So if you have a break that's anywhere past 17, it has that kinase domain in it. We want to pay attention to that. But the gene is it gets into all kinds of trouble. It can break anywhere. It could break like up front of like just downstream of exon 9. We don't really care about that. So our method has to be able to differentiate where has the break happened? Does it include the business end of this gene or not? So who are the things that it fuses with? Who are these other genes that are causing this inappropriate driving of the FGFR2 gene? And this little chart just shows you that it's really FGFR2 is that those breaks in FGFR2 are very specific to cholangiocarcinoma. So you can see that. You can see that there is an FGFR1, there's an FGFR3. They also undergo these fusions. We find that in different tumor types. And so that's not uncommon in cancer where the biomarkers tend to be sometimes at least very specific to a particular type of tumor. But in the FGFR2 case, it turns out that there's all kinds of different partners. So that little pie chart on the far end there, you can see that about 40% of the partners are this other gene called BIC1. It doesn't matter what BIC1 does. All you need to know is that when they get together, then FGFR2 is inappropriately being expressed. And then you can see there's a whole bunch of other genes that can act as that driver. And then there's a whole pile, about 35.5%, where it says other. And what has happened is we've realized over the years that there are more than 150 different partners that can do this with FGFR2, and we're finding more of them all the time. So now you start to see sort of the complexity of that landscape that we have to look at in the molecular lab to figure out whether or not there is such an event happening in a tumor. So there's lots of ways that we can inappropriately activate this FGFR2. So fusions that I just talked about, and they have to include that kinase domain. They have to be in frame and make a product. So when I say that, they can't just be sort of random so that as the protein is being made, that there's no real alphabet or open reading frame for the protein to be made. So the fusion actually has to be done in a very specific way. And it's important for the lab to be able to know that and tell the clinician and the patient that, because if it's not in frame, it's not going to respond to the therapy. The fusion puzzle, as I just said, there's lots of novel partners, so we have to be aware of that and ready to handle that. There can be some very, very complex insertions that I'm not going to bore you with because even I don't understand them all. There's a lot of really weird stuff that happens with these things. And sometimes all it has to do is lop off the end of the gene, and that itself can be oncogenic. So there's lots of different, and again, for you don't need to know all these details. I'm just trying to paint a picture of what it is that we're up against. We have a lot of methods at our disposal that we can use in the lab to ask these questions. And so the next 2 slides, I'm basically going to try to walk you through very high level some of these methods and why we would or would not choose to use them. Okay, so here are the methods. So bear with me, and I'm going to try to make this painless. Okay, so at the top, we have something called FISH. So FISH stands for fluorescence in situ hybridization. And what you basically do there is you have a chromosome and you make yourself a fluorescently labeled piece of DNA that will come and sit down on an area that you're interested in. So in the case of the first one, A, that's a break apart probe. So you have the FGFR2 gene there on its chromosome, and you make one piece that will come and sit down on one end of FGFR2 and you label it in red. And you have another piece that's labeled in green and it sits on the other end of FGFR2. So if FGFR2 is intact, not broken, then those two, red and green, will be very close to each other and they'll actually fluoresce yellow because they overlap in their spectra. So we'll get a yellow signal and we can say everything's fine. If there's a break, when the fusion happens, those two things, they separate from each other. And now you have a red and a green signal that have separated. And you can see that quite easily if you look at the cells down a microscope. The other way around is the one underneath where it says dual fusion probe. And in those cases, what you can do is you can make one probe to FGFR2, let's say the green one, and then you can make a probe that's in red to one of those partner genes. So in this case, it's to BIC1, because remember, BIC1 is one of the common partners that does this. And so in this case, when you look at a normal cell, you'll see a red and a green because they're far apart from each other. But when the fusion happens, they get together and now it looks yellow because those spectra are overlapping. So again, you can see it really easily by looking at cells. And it's a great way to do it, except with 150 partners, many of which we don't know, we can't make the red one most of the time. So that's not a good method. So that's an example of something that we need to know in the lab. Now, on the other side, we have a couple of other assays. I'm going to skip the imbalance assay. It's a wonderfully elegant method that doesn't work very well. So there's not really much point in telling you about it. And we'll just look maybe at the NGS assays. So NGS, next generation sequencing, it just means we can look at a whole lot of things at the same time. And as input material, we can either use DNA or we can use RNA. If we use DNA, we have to pay attention to the fact that we have to walk through all those, remember those thin lines that were between the exons that I showed you, how many of those? The breaks when they happen in these fusions can be anywhere in those thin lines. So if we start with DNA, we have to work our way and sequence all the way through those. And there are many, many, many thousands of bases we'd have to look at. If you start with RNA, all of those things get taken away just by the cell itself as it makes RNA, and then we have far fewer things to look at. So right away, there's a consideration that RNA-based NGS is much better at being able to identify these fusions. But even there's a couple of different ways you can do NGS, and it matters. So one of the most common ways of doing NGS is making a library by something called amplicons or PCR. So we basically have one bit of DNA that is sitting in the FGFR2 gene and the other bit of DNA sitting in the partner gene, and we simply make the product. And if those two things have fused, you'll just read right across and you'll get this thing that you can sequence and you can see it. But right away, you should be able to see that that's a problem, because if we don't know the partner, we can't make that primer. So then we can't make that product. So those amplicon-based NGS platforms are notorious for not being able to see when we don't know what the partner is. We'll miss it. And what that means to patients and to clinicians is that you run the risk of getting a false negative result, right? The fusion's there, but we can't see it. So then the other assays, which are the primer extension and the hybrid capture, they get around that, basically by allowing you to sequence right through that breakpoint without having to know what the partner is. And so those are two really the methods that people tend to use when they're after genes like FGFR2. And so here, I'll just give you very quickly what the outputs might be for these assays. Again, I'm not going to walk through all of them, but the very top one is if you have an intact FGFR2 gene, so it's normal, there's nothing wrong with it, you can see that all of those assays I've just talked about are going to do just fine. If you have it fused to a known partner gene, all the assays are going to do just fine. It's after that it all starts, the wheels start coming off the bus, because as soon as you have a novel partner, or you have something that's not in frame, or you have something that's a really weird rearrangement, a lot of these assays begin to lose their ability. And the only ones that end up standing the test of time are the RNA-based ones where they don't care what the partner is. And we call those fusion or partner agnostic. Okay. So I hope that's enough of an overview to understand why it matters, how we actually do this kind of testing, and my colleagues can certainly talk more about it in the panel. So now I'm going to talk very quickly, just in a few minutes, about what's happening in Ontario So in Ontario, standard of care, which is funded by Cancer Care Ontario through Ontario Health, is for FGFR2 fusion testing, period. That's it. That's all that is offered to patients with cholangiocarcinoma in Ontario. However, through C3, we are able to offer patients a broader spectrum of testing, and that's being done at UHN. And so here's the list, and that should look familiar to you by now because it's the same list as I showed you before. So that is what UHN is offering. And just for your information, we are using an RNA-based NGS approach that is agnostic to the partner. So we should be able to see what we need to see. And we're using a panel of about 76 genes, but we don't read out on all those genes. We just look at the ones that are in this list to keep it very specific for cholangio. And so in order to get the testing in Ontario, there's a website, and then we can direct you to that. I don't really want to have to go through all this right now, but there is a website. There is a patient attestation that has to come in, and then this testing is made available to patients in Ontario through C3. So the experience to date, we joined, I think, in July. So it's been sort of 6, maybe more. I don't know how many months. I can't count while I'm standing here. So far, we've tested 63 samples that have come through the C3 program. And out of those, we've identified 6 FGFR2 fusions, so 10%. 5 of those were reported as clinically actionable. So tier 1 means it's clinically actionable. And 1 was reported as we don't know what this means. And that's a tier 3. And I'll show you why in a second. And it does highlight again that if we didn't know what that fusion was, we might have reported 6 of them as clinically actionable, but 1 of them is not likely to respond to therapy. So here are the actual fusions that we saw. And you can see the 5 that all of them, and this is kind of small, and I'm sorry about that, but you can see they all end just after exon 17. And remember, exon 17 is the end of the tyrosine kinase domain. So they all have the tyrosine kinase domain, and then they all have a different partner on the other end. You can see BIC1 is there, but so is a whole bunch of other things. Those are the ones where the cholangio in those cases should be responding to the targeted therapy. The one on the other side, though, you'll see there's no exon 17 to be seen. The break was in a different place. There is no tyrosine kinase domain in there. It's still an FGFR2 fusion, but it's not a fusion that you would expect that would actually respond to therapy. And then just to wrap up, we actually saw other variants as well, because of course we're looking at more than just FGFR1 or R2. So we saw IDH1 and KRAS and ERBB2. These are all mutations for which there is a targeted therapy. And so the real take-home message, and I'm not going to walk through this whole pie chart, but the take-home message is that if we had done standard of care only, which was FGFR2, we would have seen 5. And instead, we saw many more than that. And so of our 63 samples that we tested, we had a 50% detection rate of variants that were in some kind of variant. 40% of those were clinically actionable, meaning there is a targeted therapy available. And half of those would not have been detected if we had done standard care alone. So I think that just sort of speaks to the value of the program. And at this point, I think I will hand it off to Greta to do Quebec. Thank you.
Thank you so much. And again, thank you for supporting the program. It's my pleasure next to introduce Dr. Greta Evaristo, if I said that right, who's a pathologist from McGill University Health Center. She is a double board certified GI and liver pathologist and specializes in hepatobiliary neoplasms, medical liver pathology and education, and directs McGill's GI and liver pathology fellowship and serves on many pathology committees. And I think we've mentioned it before, but Quebec is the only province, other than the pilot project in Nova Scotia, that is providing global molecular testing for cholangiocarcinoma patients. Thank you.
Thank you very much. So, do you want to put it back? No? Should be okay? I don't know what it does. But Okay, thank you very much, everybody. So, as announced, my name is Greta Evaristo. I'm a GI and liver pathologist. So, what does that mean? I'm the doctor that sits behind the microscope and looks at your biopsies, but I never get to meet you, which is why it's special. It's particularly special for me to be here today, so that I get to meet some of you who are patients. And so my job, Dr. Feilotter did a really good job of introducing you what we collectively try to do. So my job is really just to contextualize what our experience in Quebec has been with testing molecular pathology of biliary tract cancers. I don't have any disclosures that are relevant to this talk. So bottom right is the MUHC or McGill University Health Center, the Glen side, which is where I work, which houses one of the major portion of our pathology lab, but also most importantly houses the molecular diagnostic lab or CMDL. Which is one of the two major university centers and reference centers in Montreal, Chaume being our francophone counterpart. So in Quebec as a whole, we've sort of historically divided some of the molecular testing assignments, and relatively recently we've been assigned the sarcoma molecular testing, which actually led to originally to the development of this RNA-seq panel. But as our understanding of the molecular biology and therapeutics of cholangiocarcinoma progressed, we sort of used this opportunity to repurpose the targeted RNA-seq panel that we had developed for now testing the molecularly testing the cholangiocarcinomas. And because of that setting, it gave us the opportunity to do that under the provincial umbrella. So it's actually RUMQ reimbursed. So we have the RNA-seq panel at McGill, and that, as Dr. Feilotter explained, allows us a good opportunity to test for the fusion specifically, but also gives us an opportunity to test for some of the single nucleotide variants. And officially, it has been deployed for a little under a year. So it's our experience since May 2024. The way it is ordered on our side, so just like I said, it's accessible to any Quebecois patient who has cholangiocarcinoma. We receive these specimens from any sites. In reality, what actually happens is that it's either a pathologist who diagnoses a cholangiocarcinoma or upon request of the oncologist. They send us the tissue, they fill in this OptiLab based requisition form, which is pretty basic, as you can see, with the demographics and the indication of cholangiocarcinoma. And what I do want to stress, however, is that RNA-seq has great advantages, but it also a very sensitive assay. So what comes in has to be of really, really good quality. So we do have on the cholangio.ca website, as well as our own website, we do have the requisites for this assay. And particularly in our practical experience, we've noticed that when they are not followed, we don't get particularly good results. So there is a considerable amount of tissue that goes into the assay for it to be successful. This is sort of the repertoire that we had up until April 3rd, actually. So as of yesterday, we actually went from 550-ish genes that we would standardly report to about 1,300. But most of them are relevant for sarcomas and not so much for our topic today. So I've highlighted in red boxes the clinically relevant alterations for the cholangiocarcinoma that Dr. Feilotter mentioned, which we still have in the new repertoire and can test for. Notably absent from here are amplified genes, right? So they're rare in cholangiocarcinoma, but we've seen some cases that are HER2 amplified and can be targeted for it. So I do want to draw your attention to that, because what that means is that you need to have a complementary test for amplified-based alterations. What that means for us in-house is that if I look at this biopsy that you see on your right, you see a little bit of, I don't know if you guys can actually see my arrow. Yeah. So it's lagging. So there's a little bit of normal liver here on the right, and then you see these atypical glands that are chewing up the liver. That's the actual cholangiocarcinoma. If I look at this biopsy, I make this diagnosis, I would reflexively order the RNA-seq panel on this specimen, provided that it's of adequate quality, but also would supplement it with 2 immunohistochemical tests. We do a mismatch repair for microsatellite instability reflexively on these, and we do HER2 and IHC. And that's an important thing that I think needs to be understood by patients that do get tested with us, is that they do need to have this complementary test done in their baseline. So what we've seen so far since May 2020, in terms of the demographics, the bulk of the patients that we have tested do come from the two big universities in Montreal, so us and CHUM and the related sites. But we get about 14% of our patients that come from Quebec City and associated labs, and about a little bit over 10% that come from peripheral hospitals throughout Quebec. Very little come from Sherbrooke, and I spoke with pathologists from Sherbrooke. I think it just depends on how their ordering algorithm. In terms of the actual alterations that we've seen, unfortunately, I'll start by drawing your attention to the fact that about a quarter of these assays that we've performed so far are inconclusive. And I think a lot of this has to do with, again, this is a sensitive assay. The output is great, but the input has to be of high quality. And so, unfortunately, for many of these assays, lower RNA input, low tumor cellular count makes it such that we cannot provide a reliable answer. Another half of the patients we've tested so far did not provide any clinically relevant fusion or variant. And then there's another 21% who did get a positive alterations. 7% of all the patients we've tested had a fusion, the vast majority of which was FGFR2. And then 14% of these were sequence variants, vast majority being KRAS, and then IDH1 and IDH2 following KRAS. So I think so far this has been a successful experience for us in Quebec. I think we have been able to provide clinically relevant results to the Quebecois population. Yes, the percentages are limited, but these are not percentages. These are not statistics. These are actual patients. And so I think this has been encouraging for us to be able to provide for the province. But some of the challenges that we've seen so far, I've mentioned multiple times throughout my talk. I think the sample adequacy is something that we're hoping to be able to improve. Along through, I think, education with pathologists who do send us specimens to make sure that the specimens are adequate. Another obvious challenge so far is the fact that the RNA-seq panel does not cover copy number variants, so it doesn't allow you for some of the amplified genes. And another issue, I think, is it's just the access. We're providing this panel for the entire population of Quebec, but whether this actually reaches the entire population of Quebec is sort of the remaining question. And then in terms of how to provide our results in a timely and an efficient manner. So what's next for us? I think given the fact that we still have to complement the current assay in its form with immunohistochemistry, I think the future for us would probably be to consolidate the molecular testing. Specifically in our lab, we're probably moving towards TSO 500, which would allow us to get the readouts on the fusions and the single variants, but also would allow us to get important information from copy number variations and even TMB and MSI. So on this note, I would like to thank Dr. Andrea Gomez. You got me today, but really the mastermind behind this panel is Dr. Andrea Gomez. She's the chief molecular geneticist at CMDML and has done just so much effort to put this, to bring all these new assays into our lab, and not just for us internally, but for the entire province. And I'd like to thank also Dr. Mendelaris and Dr. Asla, who are some of the HPP oncologists who often help to contextualize the results that we get. And I didn't put it on this slide, but I would like to thank, of course, the patients and the caregivers, because that's the motivation behind everything we do.
Thank you very much. And I'll introduce the last speaker of the day. It's Dr. Faisal Khan. He's the CEO of OncoHelix that provides our molecular testing for the rest of Canada other than Ontario. He's a professor at the University of Calgary. Mistake there, probably not too problematic on the slide. He directs the Hematology Translational Lab. His research focuses on cancer genomics, transplant immunology, and immunogenetics. As I mentioned, he's the founder of OncoHelix, a leading Canadian organization that specializes in esoteric cancer genome and immunology testing. So welcome.
Thank you, Rebecca. Thank you, Leonard. Thank you, C3, for allowing us to present some of the work that we have been doing for the molecular testing for cholangiocarcinoma, first in collaboration with Insight and then in collaboration with C3. In the next few slides, I will be sharing our experience and providing some data support of what beautiful presentation Harriet has done earlier, showcasing the importance of selecting the right panel for this molecular testing. So my disclosures, today I'm presenting as a co-founder of OncoHelix Inc., but I also have a role in the public healthcare system in Alberta. I'm the lead for the transplant lab in Alberta Precision Lab and a former scientific director for molecular pathology. Okay, so like many molecular labs, the genesis of what we are going to show today, or what we have been showing in this session, has its genesis back to 2002 when the first draft of human genome sequencing became available. And what we learned from that, among plethora of information, the most discouraging finding was this: that if you randomly select any two individuals, then genetically they're 99.9% similar. Very, very discouraging, right? Then you have to look a little bit more in detail, which means this 0.1% difference is actually 3 million nucleotide difference. And these 3 million nucleotides can give you different phenotypes, different susceptibility to diseases, different immunity responses, different action of drugs, and different treatment outcomes. So pretty much the whole play is focused on these different 3 million nucleotide and mainly focusing on the other 4 aspects. And that's what OncoHelix in its translational research space as well as in its clinical diagnostic space is trying to do in Canada. Okay, so who are we? We have a very strong partnership with University of Calgary, where the pre-analytical and post-analytical workflow for any molecular immunology testing is done in OncoHelix. And we use a clinically accredited facility within the University of Calgary to do the sequencing. Okay. With a simple goal, no patient, specimen, or data need to leave Canada for the sake of diagnosis or a precise treatment plan. Overall, a strong translational research. The team has published over 335 publications, and many of them have lead to the current existence of our clinical diagnostic test. As I said, over 1 million samples have been sequenced, including over 7,500 cancer sequencing profiles. 12 research projects have been translated into clinical tests, and we have tested samples from 14 different countries so far. We have 2 clinically accredited labs. One is located in Calgary, and the other one is located in Abu Dhabi in UAE, which mainly captures the Asian markets. And we have done sample testing for over 14 countries. Okay, these are our 3 major arms: molecular diagnostics, cellular immunology, and transplant diagnostics. But for today's presentation, focusing mainly on our molecular diagnostic arm. We are one of the leading programs for comprehensive genomic profiling panels. We have 3 CGP panels, OncoHelix 1, which many of us also know as Foundation 1 panel. So OncoHelix offered the Foundation 1 panel in partnership with Roche here in Calgary. OncoHelix 2 panel, which is a TST 170 panel, that's what we are doing in partnership with C3 for cholangiocarcinoma cases. This panel is being built in our lab together with Illumina and Sophia Genetics. And OncoHelix 5, which is mainly a hemato-oncology focus panel. We also are one of the leading programs for liquid biopsy panels. So we have a partnership with Memorial Sloan Kettering and Sophia Genetics, where we offer a liquid biopsy 146 gene panel, again here in Calgary. And then we also partners with Avitia for what was known as Follow-It panel of Connexia is rebranded together with OncoHelix here in Canada. So a very wide range of comprehensive genomic profiling panel from molecular to liquid biopsies. Yeah. I'm not going to go into detail of these and quickly jump over to our experience for cholangiocarcinoma cases. And a big shout out to Taylor and his team in Insight for actually picking us. Very funny story. I had a PhD student who, after finishing the PhD, wanted to join Insight, and I wanted her to stay in our program rather than going to Insight, but she decided to go to Taylor's team. And then she turned out to be the Cupid, connected Insight with OncoHelix a year later. And this whole story that I'm going to show today came out of the connection that that PhD student has built with between Insight and OncoHelix. Okay. So far, we have completed testing of 355 samples. Overall, 410 samples patients were recruited. 13% were rejected, half of them because they have tumor cellularity less than 10%. Remaining half because we did not get enough amount of DNA RNA. So we have a pretty decent success rate. We operate with just 10% tumor cellularity with a significant success. Within that, roughly 5% of our cases are rejected when we don't have enough DNA and RNA. Again, the target biomarker was FGFR2 fusion. Sponsors earlier were Insight and now C3. This is where we stand. Around 59% of cases have some form of variant detected. Around 47% of them are clinically actionable variants, pretty consistent to what Harriet has just presented before. And within these positive cases, if we look at, around 49% have more than one variant present, and around 23% have more than one type of genomic alteration, which means they may have SNV, indels, CNVs, or fusions, but more than one type of genetic alteration is present in these patients. And if you look at the number of genetic alteration, there's a significant number who have 2 to 3 variants present. And if you look at the right-hand data, 49% of patients have SNV indels, 15%, sorry, it's a typo here, it's not an indel, it's a CNV. 15% of them have different type of copy number variation, and 11% of our patients are positive for different fusions. TSP-170 is an RNA-based partner-agnostic fusion panel. As Harriet was saying, it's a gold standard method for detection of these novel fusions. This is the overall list of genes. As I said, around 49% of them have clinically significant SNVs and indels, predominantly IDH1, as you could see here in this list of genes. And when we talk about fusions, around 10.4% of our samples are positive for FGFR2 fusion with all different type of partners that you could see in this list, which again emphasize the importance of having the right method to do this type of work. Otherwise, it ultimately is a garbage in, garbage out type of mechanism where we all, and I'm going to show some of the data that we have generated in our lab to support what Harriet and I have been showing in this session so far. Okay, so very unique study, just 3 slides. We have a multi-gene NGS panel, a 52-gene panel, which is offered by most of the labs across Canada on the name of NGS CGP. It's called the TrueSight 52-gene from Illumina. Amplicon-based chemistry require both partners to detect fusion. One partner is not enough, and we have compared it with our OncoHelix 2 panel, which is a partner-agnostic RNA fusion DNA for SNV indel detection. And this is what we have seen. Over 150 samples were sequenced for both panels. 27% of SNVs were missed in the 52 gene panel. And the reason is very simple, because it's 52 gene, not 170 gene. So it is expected that we will not be capturing all SNVs. It's not a surprise. But the main result is 94% of FGFR2 fusions were missed by this panel, mainly because it's not a partner agnostic panel. It does not have a novel fusions. And that's what it is. Only 1 out of the 17 samples that we have tested with the 52 gene panel came positive. There's more to this story which we are working right now, and hopefully in next 6 months we'll be able to share more data. Now we have also compared the panel with OncoHelix 1, which is a Foundation 1 panel, which is partner agnostic but uses DNA-based fusion detection rather than RNA-based fusion detection, and another CGP panel on a different platform. These right now, our results are showing only one method is capturing 85% of the fusions, other method is capturing 45% of the fusions. So still the other CGP methods are, at least in our experience, are not as good as TST 170, which we are offering across Canada except Ontario and Quebec. This is our requisition form. So the process is C3 enrolls patient across the country, and they submit this form, this requisition form, to our portal. Then we arrange the shipment of samples to our laboratory. And once the report is available, report is sent to C3 as well as to the referring oncologist. All these panels are also available as direct-to-consumer tests in Canada at almost 1/3 of the cost that some of other organizations like Foundation 1 are charging, minus Trump's tariff. Overall, this is the overall summary that we have completed testing of over 350 patients. 49 to 59 patients have at least one tier 1 or tier 2 genomic alteration. 10.4% patients have FGFR2 fusions. Comparison of different CGP and multi-gene methods have been done, and we firmly believe RNA-based partner-agnostic OncoHelix 2 panel has the best success in detection of FGFR2 fusions. And this is the list of all the partners who have helped and supported us in our journey so far. Thank you very much.